All right, welcome back to part three of translation. So by the end of this lesson, I hope you will be able to describe the specificity between transfer RNAs and the codon in messenger RNA. Describe the key structural features of a transfer RNA molecule. Explain how an amino acid is attached to that transfer RNA molecule and outline what we refer to as the wobble rules. So, as far back as shortly after discovery of the structure of DNA, Crick went on to propose a hypothesis of how um, translation would occur, and his idea is that transfer RNAs might act as an adapter. So he kind of proposed that recognizing a three-base codon in messenger RNA and that transfer RNA would also carry the specific amino acid for that codon. So this is kind of the adapter hypothesis. And of course, this turned out to be true. Um, Here we have a schematic of a messenger RNA with two codons. And we're showing here a particular transfer RNA, one that's specific for phenylalanine. And you can see it has the phenylalanine anticodon, and it also has phenylalanine attached to it. Over here we have um, a proline transfer RNA. You can see that it has a proline anticodon, and it also has an amino acid proline attached onto it. So transfer RNAs have a kind of cloverleaf pattern. So it has three stem loops. And if you refer back to RNA structure, remember that RNAs can fold back on themselves, forming these stem loops. There are some variable sites, and ultimately there is an extended 3' end that sticks out, and this is where the amino acid is actually put on. Now, in the following image, we are going to give you this classic cloverleaf image, but I want you to recognize that there is an actual three-dimensional structure to this molecule that isn't represented in the following image. Now, in addition to the normal A, U, G, and C that we see in RNAs in general, there are a slew of modified nucleotides that can occur. And these modifications happen after transcription. And if you can remember the end of our discussion in transcription, we talked about RNA's modifications. Um, and this is referring to one set of those. So here is that classic cloverleaf type of image. You can see there are three stem loops, 1, 2, 3. Here's the 5' end. Here's the 3' end, and you can see the 3' end kind of sticks out. This acts as the acceptor region where a covalent bond is formed with a specific amino acid. And this is carried out by a specific messenger or a specific transferase. I'm sorry, specific enzyme. Um, Down here in the middle stem loop, there is the anticodon itself. And here highlighted are some of the non-traditional nucleotides. So here we have this thing called inosine, which is actually a precursor to the purines in general. So it's along the synthesis pathway. So we make it anyways. Here we're just actually using it. But you can see we have all these different uh, derivatives of it. So Mi over here is methyl inosine, so it's methylated version of inosine. Here we can see a T. You say, wait, T's aren't supposed to be in RNAs? Well, they can be in transfer RNAs. Again, this is a modification that happens after the fact, but the end result is still the same. You now have a T in RNA. We say that you don't have T's in RNAs because it isn't put in initially. But you can see a whole slew of different versions of these out there. Now, when we look at the process of charging these transfer RNAs with a amino acid, so there's a specific enzyme called amino acid transfer RNA synthase, and there's a different one for every amino acid. So consequently, there are 20 in those rare organisms which have the 21st and 22nd amino acids. You see a specific synthase for that as well. So this requires energy. So we have a binding site for that specific amino acid that comes in there. It has a binding site for ATP. It will then couple ATP onto the amino acid. That is an energetically favorable reaction because we're releasing pyrophosphate, an energy-rich expenditure. Then we bind the transfer RNA. It will bind into the site here. At this point, we essentially replace this MP modified amino acid with the actual transfer RNA, and releasing this AMP is what provides the energy for that coupling to occur. So again, this hydrolyzing is what gives the energy for this coupling. This hydrolyzing is what gives the energy for this coupling. After the fact, you now have a charged transfer RNA. Now, something that I want you to recognize here is it costs an ATP for every one of these we make. And I want you to, as we go through the rest of translation, pay attention to how many ATPs are being used in the process of actually making proteins. This is a hidden cost, so to speak. So even making the transfer RNAs are energy rich. So when we start thinking about how we try and recover proteins and how we're very stingy on when and where we make proteins, it starts to make sense because making a protein is very energy dependent. All right. So the interaction of these synthetases with their specific transfer RNAs are kind of considered a secondary genetic code. And, you know, this has to be excessively accurate, right? You can't be putting in the wrong amino acids onto the wrong transfer RNAs, or you will end up with an incorrect protein. So these are very specific in nature, and with an error rate of less than 1 in 10,000. Sequences on that transfer RNA, and this isn't limited to the anticodon itself, is used for recognizing that specific transfer RNA. The modified bases that we see can affect translation rates, how the synthetase recognizes them, and ultimately these modified bases can be involved in codon-anticodon interactions, which we will see now. So, the wobble rule. And this is where the whole idea of having a modified um, nucleotide really comes into play. So, most of the degeneracy in the genetic code comes down to the third position of the codon. There are exceptions to this. If you recall, serine, arginine, and leucine spills over into a whole different slew in the electric or in the codon usage, but all the others where you see several codons being used for the same amino acid, it's the third base that has variability. And when you look at the structure of how transfer RNAs sit down on the messenger RNA, the positioning of that third nucleotide is not quite a perfect double helix type of scenario. So that added space gives the ability for particular codons in that third, or nucleotides in the third position of that codon, to interact in non-traditional ways. So, for example, if we take a look at the third base in messenger RNA codons, A can base pair with all of these different things, and you'll notice that most of them are modified versions. Now, U makes sense because that is the replacement for T, and you can see that there are a lot of derivatives of uracil in there, but there are others as well. Here, when we have the U in that third position right here, it can base pair with all of these. Okay. G's can base pair with all of these, and C's can base pair with all of these. And that's, again, because you don't have a perfect double helix sitting down there, it gives that nucleotide a little bit of room to flip around and interact in non-traditional base pairing forms.